Mr. President, dear ladies and gentlemen, thank you for inviting me to Iceland. This is my second visit to this country. My first visit was 4 years ago when I gave a talk in Reykjavik, and after that we received some blood samples from Icelandic ALS patients for ALS research we're doing in Sweden. I'm happy to announce that we have found the cause of some of these patients' disease. We now know why they got ALS, and I will tell you a little bit about that in my presentation, if I do not get unplugged within 45 minutes. I'll do it as quick as possible to get to the important points, but there's something I need to tell you first. See, ALS is not one disease. This condition has killed, so that is what it's doing, a number of famous people. Not only you, but it has affected a lot of people in the world. Well, we estimate there's about 38,000 ALS patients in the Western world alone. Stephen Hawkins, who's still alive for 25 years of disease, he has a ventilator. Me, by the way. David Nevin, Shostakovich, Malmstone in Sweden. This is linked to the street. Why do people get ALS? What are the predisposing risk factors? They're listed here. This is a summary. You have your family, the ground, great risk of getting the disease. Males usually get the disease often than women. Age, the older you are, the greater the risk. The top risk is about the age of 60 to 65 years. If you smoke, it increases your risk slightly. But if you have a slim body constitution, as a new research from France, you run quite a big risk of getting ALS. ALS patients are, as a group, slimmer than most of us. So if you do as what the cardiologist tells you to do, exercise, do not eat fat, you in fact increases your risk of getting ALS. So then, oh, I need to tell you a little bit. Sorry. This one as well. Farming, war veterans, physical activity may be a risk factor also, but there are slight risk factors. For women, also menopause. But what I also want to tell you is that a number of many other environmental conditions have been examined thoroughly in many different countries. We have not been able to find an association between these and the risk of getting ALS. About familial ALS, or false ALS as we call it, it's usually quoted as being about 5 to 10% of all patients report to a doctor, I have it in my family. This is a total list of all cases in the world about the frequency of familial ALS, running from about 30% in Germany to about 1% in Iceland. In Finland it is, there's no study from Iceland. On average about 5% of all patients. And in these studies, different diagnostic criteria were used. That can explain the differences in frequency. Frequently, patients do not want to tell about the family history, but they don't know about it. Sometimes, patients in the same family have developed different clinical subtypes of ALS, getting different diagnoses. Therefore, they are not considered as being familial disease, though they are in fact. Sometimes family members got the wrong diagnosis. It's no secret that 5 to 8% of all ALS patients have the wrong diagnosis. A highly sensitive issue among doctors. I'm a neurologist myself, but we got to face it. We're not very good at diagnosing ALS. You're not early this, I'll get back to that later. Sometimes the patient gets the disease very early. Just a few months ago, I had a 16-year-old girl with ALS. She never became 17. Her mother still lived, but her grandmother died in 1994 of ALS. So sometimes the children get the disease before their parents. Therefore, I can't always recognize that it is a familial condition. Sometimes the father is not the father. Also a very sensitive issue. In Sweden, we estimate that about 5% of all children do not have a biological father, they believe. Also sensitive. The figure 8% is from Italy. We do not understand reduced disease penetrance. Sometimes gene carriers do not get the disease, but others in the family have the same disease gene Gardner disease. I'll get back to that later. When we do genetic research, and I know you're very famous for this in Iceland with the company among others, what you need are the following. You need a patient. This is a very famous Swedish lady, died in 1995, and you investigate the family history, collect blood samples from family members, you compare the samples between patients and relatives, thereby you find the disease gene, then you have a disease protein, you have that, you study a protein called proteomics, you study metabolism in the disease. Metabolomics, you can make so-called transgenic mice with a disease gene, you can test new drugs in these animals, and then you can get back to the patient, testing the new drugs. This is all what we want to do. This is in fact what all researchers do if you study Parkinson's disease, Alzheimer's disease, ALS. This is the circle we all follow. And ALS research, unfortunately, we have only come to here at most. We are not still up here, in my opinion. Apologize for that. The big breakthrough in ALS research came in 1993 with this paper. This is the most important publication in ALS history, there's no doubt about it. And it is reported by Robert Brown, professor at Harvard University in Boston, United States, Nature, in March 1993. The occurrence of mutations of gene change in a gene called superoxide dismutase in familial ALS. SOD, or copper zinc SOD, SOD1 we call it for short, is a protein that looks like this, has two identical subunits, in each one we have a copper zinc atom, and SOD is a very, very common molecule. It exists in all cells, in all organisms in the world. So both Mr. Putin, Mr. Bush, elephants, malaria, mosquitoes, and tomato plants, they have in common the SOD1 enzyme. It's exactly the same in all organisms. You cannot change that. The only one who do not have a similar SOD1 are some ALS patients. And what does the SOD1 do? Normal life SOD, superoxide, is a free radical, which is metabolized, that is degraded, neutralized by SOD to hydrogen peroxide, it is then reduced to water. That is the usual mechanism taking place every millisecond in all of us here. And when SOD was found to be mutated in some ALS patients back in 1993, it was postulated by American groups that superoxide free radicals will accumulate, forming poisonous substances inside the body of neurons, killing the neurons in the ALS patient. That was the hypothesis. We later proved, using blood samples from mostly Swedish and Finnish ALS patients, but that is not the case. This is the hypothesis back in 1993, 1994, 1995. And that is the origin of the dogma that brain diseases is caused by free radicals. I'll go back to that later. This is the study we did. We collected blood samples from a large number of ALS patients from Finland, Norway, and Sweden. You have patients here in different colors from different families, compared them with family members and patients with other diseases like migraine, MS, Parkinson's disease, and healthy individuals. And you can see this is the level of the capability to reduce or remove free radicals. And the ALS patients have no real difference in the capability to neutralize free radicals. In short, you are not helped by eating vitamin E or C. It has no effect on ALS. It's an old myth. Since 1993, many research groups in the world, including ours, have been studying this SOD1 gene. We have the gene shown up here, and 130 different mutations have been found worldwide, including one in Iceland. Most of them are so-called missense mutations. The molecule, one molecule consists of 153 amino acids, one after one another. It's the order of these amino acids which determines the function of the sub-1 molecule. And 115 of those 130 mutations change this order. So there's a wrong amino acid in a specific position. 15 of them, the mutation, instead of changing the order, removes part of the molecule. And particularly we're going to talk about this one, You have G127, GGG, RQ, RW, blah, blah, blah. We call it for short G127X. That's a Danish mutation, which is by far one of the most important in the world because it removes a large part of the molecule and creating also a new sequence. And that can be used for studying the pathology of what goes out inside the brain of ALS patients with this particular mutation. This is what it looks like today. In all the countries, in Iceland, placed up here, and these are the different mutations we have found in Scandinavian countries, 14 different. And if we take as an example, we take S105L, it just tells us that the change in the molecule is in position 105, S is changed to L. In Finland, the most important mutation, number one, is marked in green, goes is found in Northern Sweden, that's in D98. The change is in position 90 from B to an A. That's just the change. You can see it as opposed to adverse. It tells us where does the damage occur. And the D98 is by far the most common SOD1 mutation in the world. Or we can also put it in another way. The most common known cause of ALS worldwide is the D98 mutation. It's very common in northern Sweden and northern Finland. And you can see in other places in Scandinavian countries, we have local areas where they have a particular family with a particular gene change. And the one we found in Iceland, 4 patients, is the G93S. That means amino acid glycine, position 93, is changed to serine. This is very, very important where you have your change. Now comes the first take-home message. Because with 14 different mutations we find in Scandinavian countries, if you go back and look at the patient who carries these gene changes, they are very different. What mutation you have determines very much what kind of ALS you get, how long you live, but also the risk of your family members, and you want to know that, usually. 6 of the mutations, listed here in red, have what we call dominant inheritance with complete penetrance. That means that if you're so unlucky to get one of these genetic changes from either your mother or your father, you will get ALS. It's a matter of time. Here we have listed other mutations, 7 others marked in blue, with so-called incomplete penetrance. That means you have inherited it, but you may not get the disease. For some unknown reason, you may not develop the disease. You can become 70, 80, or 90 years old, but you will not get ALS. But you can pass on the genetic change to your children with 50% probability. And then we have the D98, which shows recessive inheritance. I should explain. This is what it looks like. This is a typical textbook kind of ALS, where we have M is the disease gene and C is the normal gene. Remember that all genes in the body you have in 2 copies. One of them you have inherited from your father, the other one from your mother. That's the first paragraph in the law of biology. So this woman who has ALS has one disease gene called M mutated, and C is the normal or common allele. And her husband has 2 normal genes. Unfortunately, the older daughter, she gets the M, who passes on to her son, who passes on to 2 of her 3 children. And everyone who has the M gets ALS. That's what we call complete penetrance. And this is the list of SOD1 mutations who has complete penetrance. If you have any of them, you'll get ALS. I apologize for that, but that's how we cannot prevent that. That's the list today. In practical terms, it looks like this. This is one of the Swedish families from southern Sweden. We have ALS in 6 generations. This is the G114A mutation. And all patients will still have an affected parent. That means if you belong to this family and your mother or father had ALS, you will have a 50% risk of getting ALS. Or you will also put it another way, in this family, every second individual dies of ALS. And unfortunately, they are very young, between 27 and 43 years old. And they only survive just over 2.5 years. Moving to Denmark, we have a similar family. This is the G127X family. I mentioned before, where the molecule is much shorter. We have 3 siblings with ALS, and mother had the disease. Their grandmother probably had the disease, or she knew about it. But when she got pregnant again, fact, she died after self-fulfilled abortion. But her father, back in 1893, he died in the town of Lake Birger in what was called nerve inflammation, being paralyzed for just over 2 years, according to the church records. This is believed to be the oldest known ALS patient in Denmark. So that was a skipped generation, but she died young. Now moving to reduced disease penetrance, we have the same issue here. We have affected individuals with the disease, but now look at individuals marked in yellow. They carry the disease gene, but do not get the disease. We don't know why. They get old, don't they? Unfortunately, sometimes this disease will suddenly appear again. Critical terms, these are some one mutations associated with that. One of them has been found in Scandinavia, Denmark. It looks like this. This is the Danish family. We have 2 affected individuals. This man had leg onset ALS, surviving for 17 years. Initially, he got the wrong diagnosis. Fortunately, he claimed, I'm a sporadic case of ALS, I have no one in the family. My mother died of dementia at age 4. We know that. She was at the National Hospital, Hospital in Copenhagen. But her father, back in 1935, he developed difficulty with talking, swallowing, slight personality change, and he died after 15 months in late 1936, where his complete medical chart has been found. So here you have an example of a skipped generation. None of his 6 children got a disease, but his grandson got ALS. And now, recessively inherited ALS. Here we have a patient who now had 2 M's, that is 2 disease genes. In the previous example, notice there's only 1 disease gene. Recessive disease, we have 2 disease genes. One comes from the father, the other one from the mother, and only if you have 2 will you get the disease. It's pretty important. Remember that all the ones who carry 1 M now remains healthy. And it looks like this in practice. This is one of my own patients. I have 8 siblings here. One of them with ALS since 1994. He claims I'm the only one in my family with ALS. If you look at the local family, yeah, he's right. He got 1 M from his father, the other one M from his mother, and none of his siblings have developed a disease, except there's an older brother, born 1935, he also had 2 disease genes. But he's homozygous, that is, he has 2 disease genes for the D19 mutation. And this is very important because if you carry this particular mutation, you will survive for a very long time. D19 in zygous patients always have first symptom in the legs, always very slow progression, mean survival time is 14 years. Some patients have survived more than 30 years. But these patients may also show, unfortunately, a severe pain syndrome. They sometimes also get difficulty with micturition, which is very uncommon in ALS, and they may also get what we call heat sensations. This is another family. The first one was we found with D19 family, mutation back in 1993. It's on key 1 and orphan 3, where you have multiple affected individuals in different generations. And the reason being that the parents up here is that the father had ALS and his wife, they are second-degree cousins up here. So if you look into the church records, they are related. This is one of the reasons why never marry your cousin or second-degree cousins. Inbreeding is always dangerous. And this is all this is. So one mutations, unfortunately, are very rare. This is the frequency of SOD1 mutation patient with known familial ALS in different countries. This is between 12 and about 20%. And in patients with a diagnosis of sporadic ALS, it's about 0 to 7%, depending on what country you're studying. The D19, as said, is by far the most common SOD1 mutation in the world, and thereby the most common known cause of ALS is the list of countries where we found it. One study we did was in Russia. I went to Siberia year 2000 to study Russian D19 patients. We collected blood samples from different places in Siberia. These are the numbers you see here. And marked in yellow are the places, this goes to the border of Afghanistan, if I want to go there today, to get blood samples. The yellow places are the places where we find the D19 patients. They are all over the place, except they're not in Iceland. But Iceland, as a supposed to come from Scandinavia. But we haven't found anyone on Iceland with this mutation. As I said, the United States patients always have a very long survival time compared to patients with other SOD1 mutations. You don't understand that. If you have two disease genes, you should expect you will live a lot shorter. Instead, you live a lot longer than all others. I'll explain that. So what we've done a study where we've collected blood samples from all known United States patients in the world. If they live in Russia, if they live in Australia, in Canada, etc. We've studied them, and we can show that all these patients, even though many of them claim I'm the only one in the family with ALS, they're related to one another about 18,000 years ago. They had a common ancestor, all of these patients. And there we can show that patients with ALS living in Great Britain, France, Belgium, Belarus, they are one group. But from this group, someone migrated to northern Russia and Scandinavia about 1,500 years ago. And from this particular group, someone went to the Mediterranean, to France, some in France and some in Italy, about 1,100 years ago. That was probably the Vikings. Another study we've done in the United States patients is this. This is a very sensitive issue, is cognitive changes in ALS. Usually ALS is considered a disease of the motor system. It's not. This is one of the studies we've been doing, a so-called PET scanning, where we can show that the ligand substance called flumazenil, it binds to certain parts of the brain. This is from the front, this is from the left. So you have the face is here, the neck is here, the arms is down. And you can see that it highlights in the left frontal lobe in this individual who is homozygous for D19 mutation. And the interesting thing is, these changes are far more extensive than you see in healthy individuals and in controls overall. But this patient has no cognitive changes clinically. If you do a neuropsychological test, she has, but not, let's say, in day-to-day life. In short, ALS is not confined to the motor system. Other parts of the brain is also involved in ALS. Now I want to turn to the most important part of this about SOD1 and ALS, why it's so important. Because normally we have a SOD1 gene. It is inside the nucleus of the cell, in the DNA, thereby it's transformed into what we call mRNA, which goes out into the cell fluid, where the mutant SOD1 protein is created. As this is a mutation of this protein, it cannot fold normally, we believe, causing cytotoxicity inside the cell, killing the cells. That's an old notion, but if we look inside the spinal cord of ALS patients with SOD1 mutations, these are from two Danish patients with G127X mutation, who we got initial from the relative to autopsy. You can see that inside the cell body, this is the motor neuron. You can see these brown aggregates of SOD1 molecule. It sticks together the mutant SOD1 protein. This is certainly not supposed to look like this inside a motor neuron in a healthy individual. These are other pictures from the same patients. You can see the brown substance here is the SOD1 protein, which we really damage, destroys the cells. But what we have now done is a second study where my colleague Thomas Brandstrom, we've also looked at ALS patients who do not have a SOD1 mutation and looked at what does their SOD1 look like in the spinal cord. And it looks very similar. You see similar aggregates of SOD1 protein in patients with a diagnosis of sporadic ALS and in patients with a diagnosis of familial ALS. You see all the brown substance is SOD1 molecules. It's not supposed to look like it. It's supposed to be light gray like here. It's not supposed to look like this in some motor neurons. So recently, groups in Oxford, England, Lausanne in Switzerland, most recently also California, United States, tried to develop a new treatment to prevent the production of the mutant SOD1 protein. That is called siRNA, or silencing RNA. Again, we have a cell nucleus where you have the mutant
DNA, you develop mRNA, but now the new treatment is that you have small RNA molecules which can bind to the mRNA specifically, inhibiting the production of the SOD1 mutated protein. So you will not form any aggregates inside the cell. You get what we now call protein knockdown. That's modern language. It's not teenagers, it's scientists who come up with that. We call it that, thereby preventing the production of the SOD1 molecule. And the study which has been done, to show you briefly, is this. You can put this new drug, we call it, inside a virus, called so-called lentivirus. Then you can inject it by micro-injection into the spinal cord of a mouse. The spinal cord of a mouse is extremely small. It's like the size of a match. Under the microscope, you can inject this into the spinal cord, and here you have your motor neurons, which innervate the muscles. If you do two such injections in a mouse, you can show that these mice, where you do just two injections, one on either side, to hind legs, you get increased survival time. These so-called G93A mice, these are the so-called transgenic mice we spoke about before, usually get ALS around the age of 90 days and die after about 20 days. But if you give them this kind of lentivirus injections, they survive for approximately 25, sometimes 20 days more, just after two small injections. And it can be shown like this. I want to show you this moving. If it works. Of course it will not work now. I'll try this one instead. Okay, what you see up here is a mouse swimming. It's a camera below the water level. And it swims quite well. Now we have a mouse with ALS, untreated. It has difficulty with swimming. You can see that the left hind leg is paralyzed. It swims only with one leg. Mice do not usually swim with their front legs. And it's difficulty with swimming, and difficult in climbing up on the landing platform to the left. In fact, the mouse is about to drown. This difficulty with swimming. And this is, here you come, this is a sibling which has been treated with this lentivirus. It swims a lot faster and can easily climb up on the platform on the left. So this is the treated mouse, this is the untreated mouse, and this is the control mouse, which does not have the evidence. And we are planning or hoping to do a study like this in humans beginning next year or in 2008. I would like to emphasize that it is only for patients who has a SOD1 gene change, because we need to have a gene which we can knock down for this method. It's a new method which may be useful within a few years. Another study we're doing is this study, which I think Brian, you maybe talk about later. This is a study about the age of onset in ALS patients with a D19 mutation. So there are so many patients with this mutation all over the world. The youngest have been 20 years old, the oldest 94 years old, when they got the disease. And the question you, as a scientist, you ask, why does someone develop a disease at age 20, others at age 87 or age 94? They have the same disease gene, but there's a big difference in their age of onset. So we made a study where we have compared all of these patients to find another gene which will determine the age when you get the disease. In fact, we have found some evidence that chromosome 10 is a genetic change which determines, more or less like a clock, when you get the disease, not if you get the disease. That is determined by the SAT1. But there's a clock, you can say, or gene function on chromosome 10, which will determine the risk of getting the disease when you get it. And in fact, the same genetic change or area has also been found in some families with Parkinson's disease and Alzheimer's disease. The gene itself has not been identified yet. When turning to other genes resulting in ALS, some years ago, a Bergmann research group, Peter Andersen and co-workers reported in the genetics that when we looked at a gene called VEGF, vascular endothelial growth factor, and made some changes to that gene in a mouse model, these mice, to a big surprise, these were looking for cancer. These researchers, those mice got ALS instead from the change the VEGF gene. So we did a study where we looked at VEGF in ALS patients, and we found a very strong change in some patients with bulbar onset ALS, that is ALS which begins with difficulty with talking or swallowing, particularly women over the age of 70. They have quite frequently, particularly in Sweden, change in VEGF gene. And we believe that is these patients the cause of the disease. Now recently we have looked at another gene called angiogenin, and we've done the world's largest genetic study in ALS, studying about 1,600 ALS patients, looking for changes in angiogenin. And angiogenin is a molecule which is more or less accustomed to the VEGF. It's very similar in function, we believe. And 15 of those 1,600 patients have a change in the angiogenin. The remaining 1,614 did not. So you need to analyze a lot of blood samples to find just what's 15, and one of those 15 was a Swedish patient, one of them was mine. Most of the others came from Ireland or from Scotland. You can see that. So to summarize, very few patients have mutation in no disease genes. So one mutation are found in 4 to 7% of all patients. Neurofilaments and head change about 1, but the other genetic changes are extremely rare, rarely found, and we really do not know about any major gene causing ALS yet. In the last few slides, I would like to show you some of the genetic research going on. This is a Swedish family with ALS and dementia. Usually they're considered dementia and ALS different diseases. And in this family, marked in black, we have individuals with dementia, and marked in red, individuals with ALS. And textbook tells us these are different diseases. However, we've done genetic studies comparing the dementia patient and the ALS patient in this family, and have to our surprise found that they have a very great linkage to an area of chromosome 9 where there appears to be one gene which can cause either ALS or dementia in different members of the same family. Some will get ALS, some will get dementia. Why that is the case, we do not know. The gene itself has not been found. And then I'll tell you a little bit about this family. This will be my last slide, because this is the oldest known picture of an ALS patient in the world. This is Johannes Marcel. The picture was taken in Stensele, Sweden in June 1887. That was written on the back of his picture. He's sitting here with his wife and 11 of her 13 children. And he was a member of the Swedish Parliament. He was a church vicar. He was not a nobody. He was an extremely important person in Sweden in the 1880s. And if you look close at how he's holding two of his sons, he's holding them like this, not like this, as you should expect. And if you focus on his hands, you can see he has severe atrophy at his muscle wasting of his small muscles in both hands. And the following year, he died. And this is from the Book of Death, or the record of death, at the cathedral in Uppsala in Sweden in 1888. Johannes Marcel died on July 3rd at the age of 63 of progressive muscle atrophy. This is his pedigree. His that. He had 5 siblings, none of them got the disease. None of his 13 children got the disease. But if you then construct a big pedigree, this is a very compromised version of the pedigree, complete pedigree, go from that wall to that wall, you can see that marked in red are the ALS patients. Most of them have unaffected parents. There's an exception there, but otherwise, patients usually, this family has been diagnosed as sporadic ALS. But if you then extend the pedigree and include patients with dementia, in the same pedigree group called that, marked in blue, particular dementia of the frontal lobes, you will see that they aggregate close to the ALS patients. They are parents, children, siblings of patients with ALS. And if we extend the area further up in this area, where comes the family from, we now take all ALS patients from that particular geographic area, everyone, not only the one which according to the church records should belong to this parent family, but take everyone and study them. The pedigree now suddenly extends. This part here connects up here. We can show, this is the original blood cell pedigree. We can show that in this area, 38 ALS patients with sporadic ALS diagnosis, in fact, are all related to one another after here, 14 generations back. This, we believe, supports the notion that, in fact, ALS, in many cases, also sporadic ALS, is genetically. There's many different genes, we believe there are 2 or 3 genes interacting between one another. And it's the combination, the intermarriage, particularly of related parents, which causes the disease of these individuals. This is another way of showing how the patients in the muscle pedigree are related to one another. We always follow them up, now we come to another affected individual. Then, lastly, I would like to show you briefly the new drugs being tested in ALS. These are the new drugs being tested in different studies around the world. None of those studies are going on in Nordic countries, unfortunately. In the United States, they're using bone marrow transplantation or umbilical cord transplantation, being tested in Houston, Texas, and in San Francisco. We're planning to do these gene shutdown studies I mentioned before. That will be done in Los Angeles, in San Diego, probably this year, on 16 patients. And then stem cells. A study on stem cells is going on in Milano, in Italy. For some years we've treated 7 patients. This is not a clinical trial, but just trying, is it possible to introduce stem cells into the spinal cord of ALS patients? The answer is yes. Does it have an effect? Unfortunately, no. That's what we can say today. As we can talk about that later. Then, by Riluzole, I want to emphasize that the drug Riluzole, which slows down the disease progression, the effect of that is dramatically increased. I'm sure about the next. In 3 studies, and there's a 4th coming out from Canada, showing that the earlier onset of drug treatment with Riluzole, the better effect you get. We usually say you only get 2 or 3 months. That is if you introduce the drug pretty late in the disease. If you get an early diagnosis, start medication early, you can get an extension of your life by at least 12 months and even more in some studies from different countries. And with that, I would like to thank you for your attention. This is our research group. Thank you very much.